Hello everyone, Mr. Smith here, and what I would like to do today is briefly go over some topics within the genetic world, and we're going to primarily focus on meiosis today, as well as Mendelian genetics. So the question of the day is simply, how does the body create sex cells? Now, you have to remember, we're leaving the cell unit, and when we look at the question as how does the body create a sex cell, is simply centered around meiosis, which might make you recall back to things such as mitosis. Now remember, mitosis is an asexual form of reproduction. It produces somatic cells. Somatic cells are totally opposite from sex cells because somatic cells are just normal body cells, such as your skin, hair, nails, things that help you grow, cells that are there to help repair. But here, when we look at meiosis, we are simply talking about sex cells. Now, remember, when we're talking about sexual reproduction, that means that we are requiring at least two parents. The reason why I say at least two parents is because we have plants that can fertilize many other plants, and they're still sharing their genetic diversity. Offspring, and since we have more than one parent, the offspring is going to share the traits from both parents. And because we're seeing a mixture of traits, we're going to see an increasing amount of variation, which simply means we're going to see change within the population. Now, on the left side of the screen is mitosis, the right side is meiosis. The only difference here is that meiosis is going to go through these phases twice, whereas mitosis only goes through those phases once. Now, if we look at mitosis, we see that the final product is two cells. Those are our two identical daughter cells. Now remember, in meiosis, because we're going through the phases two times, we're going to divide those two cells into four. But remember, they're not going to be identical because mitosis is asexual, meaning that it's one parent and the offspring looks like identical clones. The only stages that we are going through today is going to be prophase 1 and metaphase 1 because they are the most important when it comes to meiosis. Now, before we get into prophase 1 and prophase 2, I just want to sum up your life. As humans, in every single one of our cells, we have a nucleus, and within that nucleus, we contain our DNA. Now, what encloses the DNA is something known as a chromosome. In humans, we have 46 chromosomes, meaning that we have 23 pair. The 23rd pair is going to be our sex determining chromosomes. Males have an XY, where females have an XX. And as you can see that this X chromosome, and you can see here that this Y chromosome is significantly smaller than the X chromosomes. But this Y chromosome is very, very important in differentiating the differences between a male and a female, because this Y chromosome is loaded with testosterone, which helps develop a child into a young boy. Now, when we look at the amount of chromosomes in a cell, the cell can be in different states. The cell can be in the diploid state, the cell can be in the polyploid state, or it can be in the haploid state. Remember, the prefix di means two. So that means that we have two copies. N means chromosomes. So we have two copies of chromosomes. N is going to equal to 23. 23 times 2 is going to equal to 46 chromosomes. Polyploid means that we have double the amount. So we simply just multiply this number here by 2 to get 4. That means we're going to increase that amount from 46 to 92. Haploid means that we contain half the amount of DNA. So that means that we're going to remove the 2 by dividing by 2, and that'll get us down to 23. I'm going to explain to you all why all of these numbers are important in a moment. Now, when we look at meiosis, remember we have two stages of meiosis, or we go through meiosis twice. So that's why here, compared to mitosis, you see after metaphase, you see the number 1. Why? Because this is the first metaphase. Now here in metaphase, we have what's known as homologous chromosomes. Remember, the prefix homo means the same. These are the same chromosomes, the same traits. And what's going to happen is these chromosomes are going to do the same thing in metaphase. They're going to align in the middle. The spindle fibers are going to attach to their sides. And then what you see here is that these chromosomes are crossing over. And what crossing over means is that these pieces are going to snap off through the process of synapses, and they're going to recombine DNA, and that is how genetic variation is increased. So here is just a quick little mini short version of how meiosis undergoes the process twice to allow us to end up with 4 daughter cells. Now, these daughter cells in meiosis are technically gametes because we are essentially trying to create a sex cell. I say that they are 4 unique gametes because they are genetically different due to crossover during prophase 1. Haploid because they're half the amount of chromosomes. Let's go back a slide really quick. We have 2N that doubles to 4N. We're polyploid. Then the cell splits into 2 diploid cells. But remember, we go through the phases twice. So these 2 diploid cells would then undergo meiosis, which ends us with 4 haploid, 23 chromosomes each in every single one of these nuclei. Really important to note, here in prophase 1, DNA doubles, but it does not double again once we get to meiosis 2. Now, remember, a gamete is what a sex cell is. That is a sperm and that is an egg. Males have sperm, females have eggs. But my question to you all today is, who determines the sex of the child? Is it the father or is it the mom? And just to answer that question, the father is going to be the one who determines the sex of the child. Why is that the case? Because mom only has one type of chromosome. That is an X. She can only donate an X chromosome. Remember, you give off half. Mom can give off one of these X's. But dad can give off an X, or dad can give off a Y. If the Y chromosome being carried by the sperm reaches the egg and it fertilizes the egg, the gender would be a male. But if dad's X gets to the chromosome or to mom's egg and fertilizes the egg, then the child would be a daughter. But sometimes there can be errors in meiosis. And have you ever heard the saying that people should stop having children after a certain age? And the reason for that is because the chromosomes during anaphase, where we know that these chromosomes separate, that's what's known as a disjunction effect, where chromosomes are being separated. But sometimes these chromosomes do not separate properly, and what that is known as a non-disjunction mutation. Remember, disjunction means to separate. Non-disjunction means that it is not separating, and that could cause a gamete to carry either an extra chromosome or be lacking a chromosome. Remember, you're supposed to have 2 pair of chromosomes, one from mom, one from dad, one from mom, one from dad. But here we have an additional chromosome, and because we have an extra or a third copy of a chromosome, that is known as a trisomy. If we are missing a chromosome, that is known as a monosomy, because mono means one and we only have one chromosome. Now, examples of this, the most common example would be Down syndrome, but I don't want you to think every single time you hear of a non-disjunction mutation, the result of that mutation is that of a child that has Down syndrome. Down syndrome means that you have an extra copy of the 21st chromosome. Remember, we have 46 chromosomes or 23 pair. Down syndrome means that you have an extra copy of the 21st pair chromosome. You can be a monosomy X, which means that this child is missing either an X or a Y chromosome. But an X chromosome by itself still produces a female, because Y chromosome is going to introduce the testosterone that will produce a male. But then you can be XXY, which means this is a trisomy. We have an additional X or a Y chromosome, and that is what's known as a Klinefelter's syndrome. Here we have an extra X chromosome that is super female. Now, we can detect these mutations through karyotypes. Karyotypes is normally when we just simply arrange chromosomes from biggest to smallest. But what about these chromosomes here? Because this X chromosome is definitely bigger than the 22nd pair. We always leave the X chromosome at the end of the karyotype. So all sex determining chromosomes appear at the end. So this would be a normal karyotype of a female. We have two X chromosomes here. Why do I say it's normal? We have two pair of chromosomes for each and every single chromosome number. Now, if I were to look at some mistakes, I could see here at the 21st chromosome, we have an additional copy. So this would be a trisomy 21 for a male because we have a Y chromosome present. And this would be a Down syndrome male. Here we are missing an X or a Y chromosome. So this would be an example of a monosomy X scenario. Now, because we are pushing for time, I just want to go over a couple things really quick when it comes to heredity. What do these chromosomes do when a mom and a dad unite? We understand that they make children, but we need to understand what is heredity and how do these traits get passed on. So remember, a homologous chromosome is inherited from one's mother and one's father, and they are going to be carrying specific traits. Now, what we see here inside of these chromosomes is that we have letters. Now, these letters are going to be our alleles. Now, this may be a quick little recall going back to your freshman bio, but remember, these letters represent alleles. They could be dominant, meaning that they're capitalized, or they could be lowercase, meaning that they are recessive. When we pair these alleles, we need to understand what they mean when they pair together. When you see two dominant alleles pair together, that is homozygous dominant. When you have a dominant and recessive allele pair together, that is heterozygous. Remember, hetero means different. So we see two different alleles appear together, that is heterozygous. Two of the same is homozygous. But you could be homozygous dominant or you could be homozygous recessive. Remember, if it is homozygous recessive, it is lowercase a, or it could be lowercase b, lowercase b. Now, these alleles are going to produce two different things, a genotype and a phenotype. A phenotype is a physical appearance, such as eye color. You can see that someone has blue eyes, brown eyes, green eyes, hazel eyes. But a genotype is something that you cannot see. This is the genetic makeup of an organism, such as little b, big B, big B, big B, little b, homozygous dominant, heterozygous dominant, homozygous recessive. I cannot see those things. A phenotype is a physical appearance. Now, we're going to skip this practice problem, but I did upload this PowerPoint to the website. You could practice on your own. We're going to do some work on USA Test Prep today, and I'm also going to be assigning work later on in the week. Now, I want you all to practice this work. Do not allow it to overwhelm you. Ask any questions that you may have. I understand that we're doing things online, and this is one of those assignments that it probably would have been better if we did this in person, but stay strong, y'all. AP test is coming up, and I believe in y'all. So if you have any questions, make sure that you drop it in the chat section on Teams, and y'all have a good one.